Hello everyone. So today in this current video of mine, I would be discussing about, I would be starting up with a very important major pathway for the degradation of glycogen, because as you have seen my previous videos, in continuation with those previous videos, in the earlier video we had discussed about the transport of glucose, and I simply told you that glucose is transported from the intestine to various organs with the help of the transporters, be it sodium dependent glucose transport, be it sodium independent, or you can say be it insulin dependent or insulin independent. And I told you one very important point to remember about that is the moment all the monosaccharides, glucose, fructose and galactose, they come and settle on the cell membrane, the first step should be to put a charge on them, because it is bidirectional, so that once the charge is put on them, they cannot go back in circulation. And then I told you what is this metabolism all about. So if we talk about metabolism, it is nothing but it is simply anabolism plus catabolism. So whatever energy is given to synthesize something, that energy will be given out in the degradation of something. And always keep one thing in mind, our body has different stages depending upon when we are eating, when we are not eating. And being a student of medicine, one has to understand the underlying concept that there is a well-fed state, fasting stage, prolonged fasting, which is starvation, and prolonged starvation. So the entire thing, the entire biochemistry revolves around these stages. So today we are going to discuss a very fundamental, or you can say the ultimate pathway of oxidation of glucose, which is glycolysis. Now, if we talk about, as I've been telling you from day one, that most of the, majority of these words, they are self-explanatory. If we talk about glycolysis, lysis means breakdown. Glyco is carbohydrate. So, breakdown of carb is known as glycolysis. Or you can say carb, or more specifically I told you, since glucose is the ultimate hero, a main hero in the body, so it would be the breakdown of glucose. Now the question arises, what all you have to know about any process for that matter. First of all, you should know whether it is a shunt, whether it is a cycle, whether it is a pathway. Second important thing, you should know where it is occurring actually. Because whether it is anabolic, whether it is catabolic, this we have already given you the briefing about that. So what is the site? Where is the site actually of this process? Third important point is what is the energetics? If something is being broken down, if something is being synthesized, it has to be related with some kind of energy. Otherwise, there is no point of that process. So the energetics. And the fourth, the most important point is the regulation. Because keep one thing in mind, biochemistry without regulation is incomplete. We should know at what particular step that process is being regulated. So this all we are going to cover in this glycolysis. Now since I have written it is simply the breakdown of glucose, so always remember we call it as partial breakdown of glucose. That means glucose is not completely broken down. So glycolysis is partial breakdown of glucose. Now the question is where it is occurring. So, or you can say what is the site. Now if we talk about the site of glycolysis, it is occurring in the cytoplasm. And the most important point which we should know about this is that this is a catabolic process. Right? That means energy will be given out. Okay. So, and if we talk about the energetics, it can proceed in both the mediums. It can proceed in aerobic medium, that means in the presence of oxygen, and it gives 7 ATP molecules. And if we talk about, it takes place in the absence of oxygen also, like in the skeletal muscle, anaerobic. So in the absence of oxygen, then also it undergoes some kind of energetics, producing 2 ATP. Okay. And if we talk about, again I told you about 3 things, site, then we talk about the regulation. Energetics we have already done, energetics. So if we talk about the regulation, there are basically 10 enzyme catalyzed reactions, and out of those 10 enzyme catalyzed reactions, 3 reactions are irreversible. And obviously, if 3 reactions are irreversible, they will become the regulatory reactions. The plain and simple reason behind this is, if a reaction is irreversible, we can actually regulate the entire process. Okay, so these are the 3 things which we should keep in mind before starting up with any process. This is a pathway basically. So I would say it is the partial breakdown of glucose. The site is the cytoplasm. What kind of process this is simply a catabolic process. And if we talk about the energetics, aerobically, when one molecule of glucose is broken via glycolysis, it always gives 7 ATP, and anaerobically it always gives 2 ATP. And there are 3 irreversible reactions which are actually regulating the entire flux. Then it has 3 phases, and if we talk about the phases, there are 3 phases. First is obviously energy investment phase. Whenever we have to, get a return, we need to invest something. So first is energy investment phase. Then second phase is splitting phase. And whatever energy is invested during the first phase, that is generated back. So energy generation phase. So these are the 3 phases of glycolysis. And but just keep one thing in mind, it is the incomplete breakdown of glucose or partial breakdown. So let's start up with this cycle, this pathway. So what happens is, the first molecule, because again just go back to my previous video, I told you whenever any monosaccharide is entering, it needs to be phosphorylated first. So if we talk about the first reaction or the first step, this glucose will be phosphorylated in the presence of the enzyme hexokinase or glucokinase. Obviously kinase is there. I've told you kinase always attaches inorganic phosphate. From where that phosphate is coming? From ATP. And this reaction is taking place in the presence of magnesium. So glucose is going to form glucose 6-phosphate. And if we talk about the energy investment, we can say that 1 molecule of ATP is invested. Now, this glucose 6-phosphate will be converted to, by a reversible reaction, to fructose 6-phosphate. A simple reaction is there. The enzyme will be simply, because it is just, they are the structural isomers. So the enzyme would be simply, you can say, phosphohexoisomerase. And it is so simple to actually learn the names of the enzymes as the function they are catalyzing. Now once this fructose 6-phosphate is produced, now it will undergo another phosphorylation reaction, means using 1 more molecule of ATP by the second irreversible reaction. And it is going to form fructose 1,6-bis-phosphate. Obviously, you can see that 1 means 1 more ATP molecule is utilized. So ATP 2 ADP in the presence of magnesium ions and 1 ATP, and the name of the enzyme is phosphofructokinase, and commonly we call it as PFK1, very important regulatory enzyme. On the other hand, there is one more activity which is going on. So this fructose 6-phosphate will actually undergo phosphorylation in the presence of PFK2. And always keep one thing in mind, this PFK actually, phosphofructokinase, is a tandem enzyme. If first one is acting, then fructose 1,6-bisphosphate is produced, but if 2 is acting, it will form fructose 2,6-bisphosphate. And the basic difference is that obviously we can see that PFK1 is catalyzing irreversible reaction, and fructose 2,6-bisphosphate can be converted into fructose 6-phosphate. And here the name of the enzyme is fructose 2,6-bisphosphatase, obviously digesting phosphate. Okay, and I can simply write one more ATP is utilized. So here I can say that the first phase ends, which is energy investment phase. And how many molecules of ATP are invested? 2 molecules of ATP are invested to make fructose 1,6-bisphosphate from glucose. Now comes the second phase. The second phase is splitting phase. So this fructose 1,6-bisphosphate will split up into, because it has 6 carbon atoms, so it will split up into 3 carbon atoms each. And the first product will be 3-phosphoglyceraldehyde, which has 3 carbon atoms. And since even splitting is there, so it is dihydroxyacetone phosphate, again 3 carbon atoms. And these two are linked as isomers with respect to one another. And if we have to name the enzymes, here the enzyme is splitting enzyme, which is aldolase, or which belongs to lysis class of enzymes, and here it is triose phosphate isomerase. So we can say that like this. So obviously, since they are linked as isomers, whatever is the fate of 3-phosphoglyceraldehyde will be the phase of dihydroxyacetone phosphate, right? So we can simply continue either with 3-PGA or we can continue with DHAP. This becomes the splitting phase. And now whatever energy is invested during the first phase, that has to be generated back, because in the starting only I told you it's a catabolic process. If it is catabolic, there has to be some evolution, some release of energy. Now what will happen is, this 3-phosphoglyceraldehyde will undergo again, this is again a reversible reaction in which it is oxidized, and this is the only reaction of glycolysis which depicts the aerobic, you can say, capacity of glycolysis, and it is going to form 1,3-bis phosphoglyceric acid. The number of carbon atoms are now going to remain the same. We started with glucose, which has 6 carbon atoms, and by the time splitting and energy generation phase starts, the number of carbon atoms is going to remain the same, that is 3 carbon atoms only. That is why it is partial breakdown. Okay. And here if we talk about the enzyme oxidation is there. So the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Or I can write it 3-phosphoglyceraldehyde dehydrogenase. And it is very easy to remember the name of the enzymes. Even if you don't remember the name of all the enzymes, it is not very important, but yes, the regulatory enzymes you should know, and wherever the energy is being produced, those steps you should know. And I can simply say that here the most important reaction which will take place is that NAD will be converted into NADH plus H positive. NADH plus H positive. I'll just write it in square brackets so that you don't miss this. NADH plus H positive, like this. And obviously inorganic phosphate is coming. Now, once this 1,3-bisphosphoglyceric acid is produced, now it is the time to give energy. And this will again undergo a reversible kinase reaction. And to your knowledge, this is the only reversible kinase reaction in our body. So what happens is, I can say 1,3-bisphosphoglyceric acid will be converted into 3-phosphoglyceric acid. The difference between this and this is, if we talk about this is 3-phosphoglyceraldehyde and this is 3-phosphoglyceric acid. Now you see one molecule of phosphate has been given out. Who takes this phosphate? This is taken by ADP. And ADP will be converted into ATP. So here I can say that 2 ATP will come out. So 2 ATP comes out. And if we talk about the enzyme is simply glycerate kinase. Because kinase activity is there and it is again only reversible kinase reaction. Now, once this 3-PGA is there, there is transfer of this phosphate from 3rd position to 2nd position, and 3-PGA is converted into, again by a reversible reaction, to 2-PGA. Since there is shift, so this is phosphoglyceromutase. Now, when this 2-PGA is produced, this is again a very important reaction. This 2-PGA undergoes dehydration. So, the enzyme should be dehydratase and commonly we call it as enolase, and this 2-PGA forms PEP. PEP, which is phosphoenol pyruvic acid. And here the enzyme is enolase. Right, and I can simply say water is removed or dehydration. And this is a very important enzyme. The significance of this we are going to study after finishing the pathway. And now this PEP, the last but not the least reaction, this by irreversible reaction, this is the last irreversible reaction, it forms pyruvate. Now, if we talk about pyruvate, you can't see any phosphate attached to it. PEP, this is phosphoenol pyruvate, this is pyruvate. So, obviously the phosphate goes out, and the phosphate goes out in the form of what? Again taken by ADP to form ATP, right? And I can simply say here that again 2 ATP will be produced. And coming over to the name of the enzyme, again kinase activity is there. So, it is pyruvate kinase. So, this is entire glycolysis, and if we talk about the number of carbon atoms, pyruvate is still having 3 carbon atoms, means 3 carbon atoms are still intact, only 3 carbon atom goes. So, I can simply say that one molecule of glucose, which has how many carbon atoms? 6 carbon atoms, it via glycolysis gives 2 molecules of pyruvate, which has 3 carbon atoms. So, that is why this is first of all a incomplete or partial breakdown. So, here it is justified. Okay. Now, after this is a very important point. I'll just highlight it. Okay, site we have done. Now we are going to study, after site I told you we should know the energetics. So if we talk about energetics, we will just focus on, let me just highlight the energetics part. 2 ATP here, 2 ATP here. Okay, 1 ATP here, 1 ATP here. And this NAD is also involved with energy, because automatically NADH plus H positive, wherever protons are there, whenever, or you can say whenever oxidation is there, oxidation means oxygen. So obviously we are going to consider the aerobic part. Okay. So now if we talk about the energetics, energetics has to be different, because you see it is taking place in the aerobic medium also and the anaerobic medium also. So first of all, if we talk about the energetics in anaerobic medium, so I'll just write energetics. Okay, so first of all, let's study energetics. Let's have a look anaerobically or in the anaerobic medium. So, if we talk about this, here I will just write the name of the reaction. Here I will write ATP consumed, and here I will write ATP produced. If we talk about the first reaction is glucose to glucose 6-phosphate. If we talk about the ATP consumed, only 1 ATP is consumed, no ATP produced. If we talk about fructose 6-phosphate to fructose 1,6-bisphosphate, 1 ATP consumed, no ATP produced. Then come over to the reactions where ATP is produced. So the first reaction is 1,3-BPG, matlab 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid. Here how many ATP are produced? 2 ATP are produced, none is consumed. And the last reaction is conversion of PEP to pyruvate. And here again 2 ATP are produced, no consumed. So if we talk about this, we can say this is 4 ATP, this is 2 ATP, right? And if we talk about the net gain of the ATP, we can simply say that the net gain is 4 minus 2 equivalent to 2 ATP. And that is what I have mentioned also, that in an anaerobic medium, how many ATP are produced? 2 ATP are produced. Then second is our aerobic medium. Now always keep one thing in mind, everything will be the same as anaerobic, but here 2 more NAD will be added up. So it is same as above. The only addition is of, since aerobically is there, so only addition is that when 3-phosphoglyceraldehyde is converted into 1,3-bisphosphoglyceric acid, it is producing how many ATP? Basically, or you can say NAD. So it is producing 2 NADH, which is equivalent to 5 ATP. Because here you have to understand one important thing, that one molecule of NAD always gives 2.5 ATP. Earlier it was 3 ATP, but now it is 2.5 ATP. This you have to learn by heart, because still now most of the old edition of the books, they go for 3 ATP. So I just want to rectify it. Just keep this thing in mind, one molecule of NAD will always produce 2.5, not 3. Similarly, one molecule of FAD is going to produce 1.5 ATP, not 2. Okay. So there you saw that 2 NADH, so it will be equivalent to 5 ATP. So if we talk about this net gain, first of all, total ATP are 5 plus 4 equal to 9. And here the consumption is this. So if we talk about the net gain is 9 minus 2 equivalent to 7 ATP. And that is what I have told you in the starting, that aerobically 1 molecule of glucose will always produce 7 ATP. And this you have to keep in your mind. And I'll just highlight this also because I have seen most of the students get confused between again 3 and 2.5 ka concept. So now just remember one thing, as per new concept, now though it has become old also, but still just correct that it always gives 2.5 not 3, and 1 molecule of FAD gives 1.5 not 2. So this is about energetics. Then after energetics, then we come over to the second important segment here, first of all, regulation. Now, if we talk about regulation, just now I told you that all the irreversible steps will undergo regulation. That is but obvious, quite obvious also. And if we talk about the major 3 irreversible steps, the first step here is conversion of glucose to glucose 6-phosphate. Second step is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. 2,6 also is formed, but I will tell you later. First of all, let me just write this reaction 1,6, and then the third reaction is conversion of pep to pyruvate. And if we talk about the enzymes I have already mentioned. Here it is hexokinase. I am not writing glucokinase because after that I will explain basically what is the difference between hexokinase and glucokinase. Here it is PFK1 and here it is pyruvate kinase. So now first of all we are going to take the first step, because see, regulation part you have to understand in detail, because again I am telling you, biochemistry without regulation is nothing. What we are eating, how we are eating, what happens to the foodstuffs when we are eating, this is all biochemistry. Okay. So, first of all we will take up the first reaction. Now, if we talk about the first reaction, the reaction is glucose undergoes phosphorylation to form glucose 6-phosphate. This is the reaction. And I have written 2 enzymes here. Here I have written one enzyme, hexokinase. There is one more enzyme which is acting here, which is glucokinase. Up now see ATP, chalo I will just write ATP also just to avoid confusion. ATP is utilized here. Okay. Up regulation means this is a rate limiting enzyme. If we talk about the regulation, because this I have discussed in detail in my video on enzymes. So if you have any confusion regarding rate limiting and rate committed, you can just consult that video of mine. So this is a rate limiting enzyme, or the inhibition is feedback inhibition, or we can call it as allosteric inhibition. Okay. Now see, if I talk about allosteric or feedback inhibition, I can say increased concentration of glucose 6-phosphate will inhibit hexokinase. But I am not mentioning negative sign on glucokinase. So this is the most important difference between hexokinase and glucokinase. Just pay attention here, it is very, very important. Whenever there is increased concentration of glucose 6-phosphate, it will give instructions to hexokinase. It can never instruct glucokinase. So I can say that hexokinase is under allosteric inhibition, whereas glucokinase is not under allosteric inhibition. Okay. So this is a very important difference between the functioning of hexokinase and glucokinase. Apart from this, there are other differences also, but this is one of the most important difference. Okay. And this is a rate limiting enzyme. Rate limit karta hai means that whenever there is increased concentration, it will inhibit its own formation for some time. We call it as product inhibition also. Okay. Now, coming over to this, since I have mentioned hexokinase and glucokinase, what is the basic difference between these two enzymes? Now, so hexokinase and glucokinase. And you know, there is a very one interesting feature. Glucokinase is nothing but it is hexokinase 4 only. Means it is an isoform of hexokinase. But have a look at the functions. The functions are altogether different. Now, what is the difference in the functions of this? सबसे पहले, if we talk about the site, right, then we will talk about the Km, then we will talk about the affinity, then we are going to talk about the action of insulin and inhibition. So, these are the main steps by which we can inhibit hexokinase and glucokinase. Now, if we talk about the site of hexokinases, it is present everywhere. So, all tissues. But if we talk about glucokinase is present only in the liver. It is not present elsewhere. It is only present in the liver. If we talk about Km, hexokinase has low Km. So, obviously difference hai. So, glucokinase will have high Km. And I have already discussed in my enzyme video that Km is inversely proportional to the affinity of the enzyme with the substrate. So, if any enzyme has low Km, affinity is high. Glucokinase high Km, low affinity, right? Now, coming over to the most important point, insulin. Always remember, insulin will never act on hexokinase, will not act, no action. Whereas glucokinase survives on insulin, basically. So, insulin will act on, so action is there, action present. And if we talk about inhibition, hexokinase allosteric inhibition, and here no inhibition. So, these are the very important differences between hexokinase and glucokinase. This you have to learn by heart. Very important. Now, if I talk about, जैसे I have mentioned insulin here. Okay, so what happens is whenever, and moreover, देखो, since glucokinase is present in the liver and insulin is acting on it. So, whenever we are taking a carbohydrate rich meal, automatically the concentration of glucose is going to increase. There will be a spike in the carbohydrate level in the liver. So, at that particular moment, this glucokinase will be acting. So, glucokinase karega kya? It will simply increase the uptake of glucose by the cells, and that is what is the function of insulin also, isn't it? And that is why whenever the person goes into hyperglycemia, immediately a shot of insulin is given. So, what is the, funda behind giving that shot of insulin? Insulin immediately will trigger the uptake of glucose by the cells. Okay. So, I'll just mention it here that glucokinase is present in the liver. It has very low affinity and it will act only when the concentration of glucose exceeds 100 milligram per dl. Okay. And in my, again, video on enzymes, I had clearly demarcated between their mode of action. Matlab hexokinase and glucose are like best friends forever. Whereas glucokinase and glucose are like enemies, but they have to work together. This is, suppose if an assignment is given to the enemies also, okay, to your enemy, automatically you have to submit the assignment. Whether you cry, whether you laugh, whatever is there, you have to submit that assignment. Same is the case here. So, always remember, hexokinase can always act. Hexokinase can act on glucose when the concentration is even 0.0002 milligram per dl. Any more. Or this is also known as minimal basal uptake because this is present in all the tissues. And glucokinase can act on glucose only when the concentration exceeds 100 milligram per dl in the liver. So basically glucokinase acts as a clearing factor also. Has a clearing factor. Very important. Okay. And this is the basic difference between hexokinase and glucokinase. And hexokinase की fourth form is known as glucokinase. Right. So this is about regulation, and you can say that it is the rate limiting enzyme. Just keep this thing in mind that hexokinase, or I would say it is the first rate limiting enzyme because this is the first reaction. Clear? Then moving on to the second reaction. Now if we talk about the second reaction, the second is this fructose 6-phosphate is forming fructose 1,6-bisphosphate. Here the enzyme is PFK1. And again I'll just write the entire reaction. ATP to ADP, one molecule of ATP is utilized here. Okay. And this fructose, on the other hand, this is also one of the another reactions, that fructose 6-phosphate in the presence of PFK2 will form fructose 2,6-bisphosphate, which will be converted into fructose 6-phosphate by fructose 2,6-bisphosphatase. Okay. Now coming over to this reaction, first of all we will just write, ek to this is the second rate limiting enzyme. So it is rate limiting. Why rate limiting? Because same thing. Increased concentration of this will give instructions to PFK. Similarly, it will be inhibited by ATP. It will be inhibited by citrate ions, and it will be accelerated or activated by AMP. Okay, this is very important. So I can say that ATP and citrate ions are going to inhibit PFK1. Similarly, AMP, which is a positive allosteric modulator of this enzyme, it is going to accelerate the activity of this enzyme. It is rate limiting. Now the second important point about this is the only rate committed enzyme. Rate committed enzyme of glycolysis. And it is also known as bottleneck. जैसे we cannot open the bottle without opening the cork. Same is the case with this. We have to open the cork. We have to make this reaction happen. Then only glycolysis will proceed at a much smoother rate. Okay. Now the question is, what is the difference between rate limiting enzyme and rate committed enzyme? This I have already explained in detail in my enzyme video, but here I just want to give you a brief summary that rate limiting enzymes are the ones which are an example of feedback inhibition. The moment the concentration of product increases, they start inhibiting its own production. But whenever we talk about committed mane, this fructose 1,6-bisphosphate is actually committed to undergo only and only glycolysis. Whereas if we talk about glucose 6-phosphate is not committed only to glycolysis, because that is a mandate for the cells to accept glucose. So it can undergo glycogenesis, it can undergo HAP shunt and so many other pathways. But fructose 1,6-bisphosphate will undergo only glycolysis. This fact you have to understand, because most of the students get confused actually between the concept of rate limiting and rate committed enzymes. So this is the first and the only rate committed enzyme of glycolysis, and I will just highlight it also, or the bottleneck. Ab here one very interesting process is going on. The process is there is a fight between PFK1 and PFK2. Because you know one very interesting thing, glycolysis is occurring in the fed state, okay. Gluconeogenesis is occurring in the fasting state, but in RBCs, this glycolysis is occurring in the fasting state also. So there has to be a parallel process, means if the genes, or you can say if the process which is degrading glucose by glycolysis is occurring every time, all the time, there has to be a process, antagonistic process, which when it is not taking place should give rise to glucose, that is gluconeogenesis. So in gluconeogenesis, first of all, the irreversible step is made reversible, and this fructose T6-bisphosphatase also has a very, very important role to play. So that is what I am just going to explain you here. So what happens here is, this PFK2 is activator of PFK1. So I'll just write it here that this PFK2 fructose 2,6-bisphosphate PFK2. So this is actually activating this PFK1. So what will happen is fructose, or you can say it will lead to more glycolysis. Right? On the other hand, this is glycolysis. Now what happens is reverse is happening in gluconeogenesis. So in gluconeogenesis, this fructose 2,6-bisphosphatase, I'll just write it here also, fructose 2,6-bisphosphatase is the name of the enzyme. This is going to inhibit PFK, thereby decreasing glycolysis. So if glycolysis is decreased, so automatically fructose 1,6-bisphosphate won't proceed further. Instead, fructose 6-phosphate will be there, and it is going to increase gluconeogenesis. This is a very important concept. Okay, so I can simply say that fructose 2,6-bisphosphatase is actually inhibiting PFK1, whereas PFK2 accelerates PFK1 so that more and more glycolysis takes place. And I'll just highlight it like this, very, very important. And that is why we say fructose 2,6-bisphosphate is actually acting as an intracellular energy indicator of the body. Intracellular energy indicator. Of the body. And that is why this is also known as the bottleneck of glycolysis. Then we come over to the third step. Third step is a very simple step. Simple. Phosphoenol pyruvic acid is converted into pyruvate. ATP is produced from ADP, and the name of the enzyme is pyruvate kinase. And if we talk about the properties of this enzyme, this is the third rate limiting enzyme. So it is simply rate limiting. Limiting enzyme. So the mode of inhibition is feedback inhibition. Inhibition, or I can say that increased concentration of pyruvate will start inhibiting its own synthesis. So this is the third rate limiting enzyme. Okay. So pura regulation, if we see in detail, I can simply say that there are 3 rate limiting enzymes and 1 rate committed enzyme. The 3 rate limiting enzymes are hexokinase, phosphofructokinase 1 and pyruvate kinase, and the rate committed enzyme is only one, that is phosphofructokinase 1. So this is the regulation. Now we have to regulate on the level of the hormones also. So ये तो regulation क्या था? It was simple, feedback inhibition. Second regulation is hormonal regulation. Now, if we talk about the hormonal regulation, the most important point which we have to remember is that insulin always promotes glycolysis, whereas glucagon always inhibits glycolysis. So I can say that in the body, when there is high insulin to glucagon ratio, glycolysis will be accelerated, and if there is low insulin to glucagon ratio, glycolysis will be decreased. And slowly as we are going to proceed further in metabolism, this terminology will actually be clear to you. What is the difference between insulin, the mode of action of insulin and glucagon? And both are released by the pancreas itself. Jaise glucagon we know it is released from the alpha cells, insulin is released from the beta cells, and these two hormones play a very important antagonistic role in the metabolism of carbohydrates and obviously in fats also. But here we are studying carbs. Okay, so this is the regulation on the level of the hormones. And one more very important thing, we call insulin as an anabolic hormone, isn't it? Because insulin is released in hyperglycemia, it leads to hypoglycemia. Glucagon is catabolic hormone because it is released in hypoglycemia, it brings the body in hyperglycemia. But this is the only exception in the body, that insulin being an anabolic hormone, but still it catalyzes glycolysis, which is a catabolic process. On the other hand, glucagon being a catabolic hormone will always accelerate gluconeogenesis, which is an anabolic process. So there are certain exceptions in the body just to maintain homeostasis. Okay, so this is regulation. Then after regulation, we come over to another very important point here, substrate level phosphorylation, which is actually asked in the exam also. It is very important. Now, what do you understand by the term substrate level phosphorylation? Just now we saw there were total 4 ATP molecules which were produced. And if we clearly look at the reaction, we can simply say that the ADP is getting that inorganic phosphate from the substrate, and that is what exactly happens here. So substrate level phosphorylation is attachment of inorganic phosphate at the expense of the substrate. Expense of the substrate. Because phosphorylation is attachment of phosphate, but when that phosphate is coming from the substrate, it becomes substrate level. And there are only 2 reactions in glycolysis. And what are these 2 reactions? One reaction is conversion of 1,3-bis-phosphoglyceric acid into 3-phosphoglyceric acid, synthesis of ATP. And the second reaction is conversion of PEP to pyruvate. Again, ADP to ATP. Here again glycerate kinase is the enzyme. Kinase will automatically come where phosphate is being attached, and here it is pyruvate kinase. So this is substrate level phosphorylation. After this, then we come over to another important aspect of glycolysis, Rapoport Lubring shunt. Rapoport Lubring shunt. Now see, Basically, you should understand what is the meaning of the shunt first of all. Shunt is that which does not have any end product. So, what is a shunt? Shunt does not have an end product. Does not have an end product. Right. And second important point is that if we talk about glucose, we know that 90% of glucose obviously is undergoing glycolysis. अभी जो 10% ग्लूकोज है ना, this 10% glucose undergoes this Rapoport Lumbering shunt in the RBCs. Only 10%. Ab 10% is again, if it is occurring in the body, it is quite a lot. Now the question is, what is the significance of this Rapoport Lumbering shunt? Why it occurs? Right. So what happens here is, first of all, let's look at the shunt. Shunt ka matlab no end product is there. Now just go back to glycolysis. In glycolysis, I told you that 1,3-bisphosphoglyceric acid is being produced. Bisphosphoglyceric acid. Okay. And it is obviously undergoing substrate-level phosphorylation, the reaction which we just finished. Or I can say ATP is being synthesized here. And this 3-PGA to 2-PGA, and like the glycolysis continues. Up this 10% of glucose is entering in the Rapoport Lumbering shunt. So what is happening there? This 1,3-bisphosphoglyceric acid will be converted into 2,3-bisphosphoglyceric acid. 2,3-bisphosphoglyceric acid. Matlab, first position se this phosphate will come to the second position. And the reaction is mutase. I am not writing the name of the entire enzyme. Simple, it will be 1,3-bisphosphoglyceric acid mutase. So even if you write mutase, you will get good marks, full marks. Now, this 2,3-BPG simply will be converted into 3-PGA. In the presence of water, it will give this inorganic phosphate. Or I'll just write hydrolysis mein it is going to give this inorganic phosphate, and that is what is our target. What is our target? That in RBCs, this 10% will not lead to the synthesis of ATP. It will simply form 2,3-BPGA, which will be converted into 3-PGA, and glycolysis will continue like that. Energy to mil gaya, but the only thing is that 2,3-BPG is produced. So if I have to simply say what is that RLS? RLS is this 1,3-BPG to 2,3-BPG to form 3-PGA like this. Now the question arises that what is the significance, right? Because the body is taking 10% of glucose to form this 2,3-BPG. Now the significance of this Rapoport Lumbering shunt is the most important one in hypoxia. Very, very important. What happens is hypoxia, or whenever we go to any high altitude, whenever we go to the mountains for trekking, we start breathing, the breathing pattern also changes. Why? Because the level of oxygen becomes low there. So we have to put more thrust so that more oxygen should be unloaded to the tissues, and so that we can survive those anoxic episodes. So basically, what is the biochemistry behind that high altitude? There is a physiological process also which is going on. Again, we are going to study this in detail in physiology. But as per biochemistry, what happens is, in hypoxia, during hypoxia, when the level of oxygen decreases, this RLS increases. Now, if RLS is increasing, what is increasing? This 2,3-bisphosphoglyceric acid will increase. And the basic function of this 2,3-BPGA is that it starts unloading maximum oxygen to the tissues. So, unloading of oxygen to tissues increases, and that is what is the body demands, because the body is demanding more of oxygen. So always remember, in response to hypoxia, RLS will increase, leading to, because there will be a shift in the hemoglobin oxygen curve, or it will keep this hemoglobin in that taut state, because we have studied that again in physiology. The physiological phenomenon will be the curve will be shifted towards right, or I'll just write shift of curve towards right. Okay. Or there is an increase in the taut state of hemoglobin. So 2,3-bisphosphate glyceric acid will increase, and it will lead to the person can survive the anoxic episodes. Anoxic or hypoxic episodes. Very important. So I will just write here, unloads maximum oxygen to tissues. Now the question is, if any shunt, any pathway is going on, there has to be an energetics attached to it. What is the energetics of RLS? And you will be surprised to know that if we talk about the energetics, the net gain of ATP is 0 here. Energetics is because this reaction is bypassed, 1,3-BPGA to 3-PGA, where 2 ATP were formed. Okay, or you can say, I will just write the first reaction, PEP to pyruvate. Here 2 ATP are there, right? This reaction does not take place here. So if we talk about the net gain would be 2 ATP produced minus 2 ATP consumed. So it becomes 0. So we can say that in RLS, even though the net gain is 0 ATP, still it provides to be a boon to survive during anoxic or hypoxic episodes. So this is a very, very important shunt. Then after this RLS, then we come over to another important point. What are the inhibitors of glycolysis? Now, if we talk about the inhibitors, the most important inhibitor which you should actually learn by heart is, I'll just make a chart also, name of the inhibitor, enzyme which is inhibited, and the function. So as such, if we talk about the name of the inhibitor, will take fluoride. Fluoride ions. Fluoride ions are the inhibitors of enolase enzyme. And the most important function of fluoride is estimation of glucose. And the second inhibitor is arsenate, or we can say iodoacetate. Arsenate is an inhibitor of glycerol-aldehyde 3-phosphate dehydrogenase. As such, function we won't say it has a very important function to perform, but yes, it is an inhibitor. Okay. So now the question is, what is the function of these fluoride ions? This is very, very important and asked in the exam also so many times. So what happens is, whenever this I have discussed in my video on enzymes also, but here also I want to focus is because this is a very important point. So what happens basically is, whenever we have to estimate glucose, estimation of glucose, सबसे important in the serum, it is done in the gray vacuotenors. And what is the summary behind these gray vacuotenors? Because they already consist of an anticoagulant. Anticoagulant can be citrate, it can be potassium oxalate, any anticoagulant will work. But along with this, it consists of sodium fluoride, because fluoride inhibits enolase enzyme. And what is the function? Function is to stop glycolysis. Because we know that life of RBCs is 120 days. So automatically, the technician is taking the sample to the primary health center, so automatically glycolysis will keep on going. And if a person is diabetic, so obviously it matters a lot, the concentration of glucose. So immediately sodium fluoride is added along with an anticoagulant. So he can actually take the sample and then he can simply, you can say, procure the sample and he can actually go for the estimation of glucose. Clear? So it is stopping glycolysis. So this is the inhibition or the inhibitors of glycolysis. Then we come over to another very important aspect in glycolysis, that glycolysis do occur in the cancer cells also. And what happens here is, actually it is known as Warburg's effect. And what is this Warburg's effect? It is simply known as aerobic glycolysis. Very, very important. Which becomes again the basis of that PET scan and all. Aerobic glycolysis. So, see, there are two cells. If I talk about a normal cell here, and if I talk about a cancer cell here, both will require glucose for their energy, obviously. Okay. So what is happening is, if I talk about a normal cell, I can simply write the process as glucose to glucose, 6-phosphate, chalo I will write, and I will simply go for PEP, or I can write simply pyruvate. This is the reaction which is going on. On the other hand, in the cancer cells, same is the process, but the only difference is that this pyruvate will be forming lactate. Very important. Okay. So if we talk about this pyruvate to lactate, again, this is going to occur in the presence of oxygen. So I can say that in normal cell, what is happening is, this is the cancerous cells. Here normal cells mein pyruvate is there, but here lactate will be there. Okay, so this is actually aerobic glycolysis. And if we talk in general, I can simply say that this pyruvate has to undergo TCA cycle. If we talk about the energetics, so it is going to produce total 32 ATP molecules. But if we talk about the cancer cells mein lactate will be produced. And if we talk about the energetics, it is going to give 2 ATP molecules. And if we talk about the enzyme here, this enzyme pyruvate kinase. Here also the enzyme is pyruvate kinase. The basic difference between these two pyruvate kinases is that this enzyme is a tetramer and it has more activity. Whereas this enzyme is a dimer and it has less activity. But at any case, cancer cell hai, cancer ko bhi to survive karna hai, cancerous cells ko. So how they are going to survive? They are going to survive on this particular metabolism. So you can see the difference in the energetics also. Now what is the significance of this whenever we carry out the PET scan? Okay, PET scan mein this method is the one which is actually used. So if we talk about the bases, what is the biochemistry in PET scan? We always use FDG, fluorodeoxyglucose. Deoxyglucose. Right, fluoro is attached. So this fluorodeoxyglucose, when it is given, it will go and increase the uptake by the cancer cells, or whatever glucose is present in the cancer cells, and which will be detected by the screening. Because there will be, again, PET scan stands for positron emission technology. Okay, so this is the basis behind that. And that is why this effect is also known as Warburg's effect, because there is aerobic glycolysis. Again, I am not going to into that much detail, but yes, you should know कि cancer cells में what is the phenomenon, what is the funda there? They undergo Warburg's effect, which is simply aerobic glycolysis. You can write Warburg as U, or since it is a noun, we can write it as WARBURG apostrophe S, or it is BERG apostrophe S. So here pyruvate to lactate is produced, and here pyruvate undergoes this 32 ATP molecules, right? So this is all about glycolysis. And if I have to summarize, I can simply summarize glycolysis as one molecule of glucose having 6 carbon atoms via 7 ATP molecules aerobically, producing 2 molecules of pyruvate, which has 3 carbon atoms. So here we end up with glycolysis, and the most important point which you have to remember here is, if we consider the rate limiting enzymes, there are 3 rate limiting enzymes. If we consider the rate committed enzymes, there is only one rate committed enzyme, which is actually the bottleneck also of glycolysis. And in glycolysis, we can correlate it with Rappaport Lubrin shunt also, where there is no net gain of ATP molecules, but still Rappaport Lubrin shunt plays a very, very important role in hypoxia. Hypoxia, or you can say high altitude physiology, also it plays a very important role. And third important point is, fluoride is inhibitor of enolase enzyme. Whenever we have to estimate glucose in serum, we always treat the sample with an anticoagulant along with sodium fluoride, because there are different type of vacutainers. Gray is there, red is there, purple is there. Every vacutainer has its own significance, but for glucose estimation it is always gray vacutainer. The reason being, it has sodium fluoride, which is an inhibitor of enolase enzyme. Then there are 2 reactions which are of substrate-level phosphorylation. One is conversion of 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid. The second reaction is conversion of phosphoenol pyruvic acid to pyruvic acid. So overall we can say 4 ATP molecules are generated, 2 at every step. Now then, after this, in the next video of ours, we are going to study what is the fate of these 2 molecules of pyruvic acid which are produced, because again this is a partial breakdown or incomplete breakdown, right? And since we are talking about oxidation, so 7, if we say anaerobic, it is 2 ATP molecules. So I hope this glycolysis is clear to all of you. Thank you so much.